Hi everyone. Thanks for your interest in this presentation. Also, many thanks to the organizer for giving us the opportunity to present our work. Today, I would like to introduce to you how our immunoglobulin humanized mouse platforms can advance therapeutic antibody discovery. Biocytogen is a biotech company that provides integrated technology platforms. Our headquarters is in Beijing. It has over 1,000 employees worldwide, including Boston site, which was established in 2018. Over the past 10 years, we have worked with more than 1,500 academic and 200 industry clients on 4 areas: gene editing, genetic modified models, antibody discovery, and preclinical studies. These 4 platforms are integrated to form a comprehensive power engine that have facilitated the success of hundreds of antibody discovery projects. For every stage of the antibody discovery process, Biocytogen provides a set of mouse models that are specifically designed. For antibody hit generation, we have immunoglobulin humanized mouse, RenMab, RenLite, and RenMab HITS. For lead optimization stage, we have target humanized mouse model. And for candidate selection stage, we have immune system humanized mouse models. Why do we use immunoglobulin humanized mice? The force of natural evolution determines the intrinsic complexity of our immune system that can be surpassed by in vitro design. In immunoglobulin humanized mice, the in vivo selection, differentiation, and affinity maturation cannot be recapitulated in vitro. The well-evolved immune system generates in vivo antibodies with less immunogenicity, high antigen-binding affinity and specificity, and also better biophysical properties and better developability. The immunoglobulin humanized mice carry full human immunoglobulin repertoire, which gives the human-like epitope and sequence diversity. All these benefits brings a shorter development timeline and higher clinical translation. Biocytogen offers 2 immunoglobulin humanized mouse models. First one is RenMab, which carries full human heavy chain and kappa light chain variable domain. And second one is RenLite, which carries a single common human kappa light chain while maintaining the full human heavy chain repertoire. RenLite is designed specifically for bispecific or multispecific antibody discovery. RenMab mouse is the first generation of our immunoglobulin humanized mouse platform. In RenMab mouse, the 2.6 megabase mouse heavy chain variable domain gene was replaced in situ with full human VDJ loci, which is 1 megabase that includes all 123 to 129 V genes, all D genes, and J genes. In light chain, the whole mouse kappa chain VJ segments was replaced in situ with full human kappa light chain VJ loci, including both the distal and proximal V clusters. The lambda light chain is undisrupted and mouse constant region is maintained to ensure proper B-cell development. During this COVID-19 pandemic, like many other biotech companies and pharmaceutical companies, Biocytogen has initiated a campaign of generating neutralizing antibodies against the SARS-Cov2 virus. 2 groups of RenMab mice were immunized using different immunization strategies, protein immunization and DNA immunization. And 2 discovery platforms were used, hybridoma and single B-cell cloning. This campaign generated a rich list of high-affinity binders, among which 8 hits were listed as example here. These 8 antibodies not only blocks the spike in human ACE2 interaction, but also have high affinity against RBD domain. These hits also successfully block pseudovirus infection of human ACE2 expressing cell line and human ACE2 transgenic mice. Not only do these antibodies functionally neutralize the SARS-CoV-2 virus with a high affinity, they also showed high epitope diversity. Synergetic assays showed that clone B2 and B5 recognize distinct epitopes within RBD domain. When used together, they inhibit virus infection more dramatically. Therefore, these 2 clones can be used as cocktail treatment for COVID-19 patients. These antibodies also showed solid drug-like properties as the thermal stability measured by DSF, hydrophobicity measured by HIC-HPLC, and isoelectric point by IEF. Biocytogen has a very strong gene editing technology platform. This makes it possible for us to further engineer the RenMab mouse. Here we introduce to you the second generation of RenMab platform, the RenMab HITS platform. There are two purposes of HITS platform. One, to break immune tolerance for challenging targets, such as high homologous proteins between human and mice. The RenMab HITS mice can generate antibodies that recognize broader epitopes as well as antibodies that cross-react cross-species to expedite clinical development. The second purpose is to assemble hundreds of knockout RenMab mice for promising therapeutic targets and provide this resource to the community and facilitate therapeutic antibody discovery. In our RenMab website, we have a RenMab HITS platform page. Here is the status overview of the target knockout mice. Right now, we have about total 600 HITS mice calculated by target, and 11 of them have been exclusively licensed out. You can search your target of interest in here and see which stage it has reached. We welcome partnership at any stage of the development. Here is the flow titer of RenMab HITS mice compared with RenMab mice. These two groups of mice were immunized with the same antigen using the same immunization regimen. We can see that at the 1 to 10,000 dilution point, RenMab HITS mice have a much higher MFI compared to the RenMab mice. This data shows RenMab HITS mice has much more robust immune response once the immune tolerance is broken. In this slide, we compared the yield of RenMab HITS with RenMab mice for cross-reactivity antibody project. As these figure shows, target B and target C each have 61% and 86% homology of their ECD domain between human and mice. RenMab HITS mice in this campaign generate higher number of positive antigen-specific B cells compared to RenMab mice. When analyzing antibody hits that were generated from these two campaigns, we found RenMab HITS mice generate much higher proportion of cross-species reactive antibodies compared to RenMab mice. Now I would like to introduce to you the common light chain RenLite mouse platform. RenLite mouse is designed for advancing bispecific and multispecific antibody discovery and solving the common issues of the bispecific antibody discovery process, immunogenicity and chain mispairing. Natural in vivo selection affinity maturation using human heavy chain repertoire reduces the immunogenicity of the bispecific antibodies. At the same time, it allows an efficient construction of different heavy chain variable domain that share the same light chain. Therefore, RenLite mice can save time and effort by avoiding the later stage intensive engineering to ensure the right combination of the heavy and light chains and reduce immunogenicity. Here is the illustration of RenLite mice humanization. First, the heavy chain, similar as RenMab. Whole mouse heavy chain variable domain was knocked out and replaced with entire 1.0 megabase human VDJ segments in situ. For light chain, whole mouse kappa VJ segments were knocked out and replaced with a single human VJ gene in situ. We select human V kappa Germline 3-11 as common light chain after thorough consideration. First, based on the distribution and germline usage of heavy chain light chain pairing in natural human immune repertoire, 3-11 is one of the most used light chain. Also, based on the drug-like property assessment and canonical structure analysis. Lastly, based on productivity assessment in clinical assets and various antibody discovery campaigns. RenLite mice has common human light chain and a full human heavy chain variable VDJ segments. With the fixed light chain gene, B cells from the RenLite mice still developed a full human diversity repertoire. As this data shows, all 46 functional V genes showed up and 25 D genes and 6 J genes showed up. The CDR analysis of antibodies derived from RenLite mice also showed a very human-like CDR3 length and amino acid composition patterns, such as the length-dependent tyrosine usage. So how does the common light chain function in the development of B cells? Since fixed light chain will not provide much variety during in vivo selection and affinity maturation, one would assume that the common light chain will undergo somatic hypermutation during this process. We tested the hypermutation rate in naive RenLite mice. Here we can see that near 75% light chain has no mutation and about 20% has one mutation. As a result, totally 95% of light chain has one or no mutation. The majority of the B cells from RenLite mice use the kappa common light chain instead of lambda light chain. How does RenLite compare to RenMab? Here we have a case study of cross-comparison of RenMab and RenLite platforms. We picked a type 1 transmembrane protein Y as target, and 3 groups of mice, wild type, RenMab, and RenLite mice, were immunized side by side with the same antigen, same schedule. The figure on the right shows the titer of all 3 groups of mice. As you can see, the titer are pretty comparable among these 3 groups of mice. After immunization, the positive mice were sacrificed and moved forward with single B cell sorting. We recently obtained a new Beacon Optofluidic system. Here is the workflow of Beacon sorting. The B cells were enriched and loaded onto the chip of the Beacon system. Within 1 day, all antigen-positive B cells were identified. These B cells were then sequenced, and the heavy and light chain variable domain sequences were extracted and validated. There are several advantages of this workflow. Firstly, it speeds up the discovery process dramatically. From immunization to getting the antibodies, it would take only 3 months. The antibody sequences were obtained at early stage, so loss of positive clone is avoided. Lastly, the antibody secreting B cells already undergo affinity maturation in vivo, so the antibodies derived from these cells have very high affinity. Here is the yield we got from this case study. You can see from this table that RenMab and RenLite mice have relatively high harvest rate of antigen-positive B cells. Normally, the sequence extraction rate is more than 70%, so for a normal target, we can identify average several hundred hits. In this study, we also analyzed the germline usage of the antibody hits between RenMab and RenLite mice. In this particular campaign, IgHV 3-30 and 1-18 are dominant in RenMab mice, and IgHV 330 and 321 are dominant in RenLite. This data indicates that RenLite, although carries a single light chain, can still utilize a broad repertoire diversity. How is the affinity of RenLite mice? Since currently we do not have the data from the same campaign, here are 2 affinity plots from 2 campaigns with different targets. You can see here that RenLite mice generate hits that has a broad range of affinity, and RenMab mice consistently shows a narrower range where majority of the hits have affinity higher than 10 nanomolar. With the fixed light chain, B cell will inevitably rely on heavy chain variable domain to generate specific antigen binding, which means affinity will be effective to some degree. Here we show affinity analysis of antibodies derived from RenLite mice. This is an antibody discovery campaign for well-known immuno-oncology target. Here is a list of antibody hits we got from this campaign. You can see that the RenLite mice generate antibodies with wide range of binding affinity, from sub-nanomolar to micromolar. A wide range of affinity provides flexibility on building antibody formats that require different degree of binding affinity. Nonetheless, for some antigens, RenLite mice can generate high affinity binders. Just like this campaign, RenLite mice generate high affinity binders against target G. Some of these hits outperforms the benchmark positive antibodies in the market. The epitope binding analysis of RenLite antibodies from another campaign indicated that here there are 9 different epitopes grouped into 4 clusters. This phylogenetic tree figure shows that these antibody hits bind to diversified epitopes. The second case study I will show you today is cross-species antibody generation using RenLite Platform. In this campaign, RenLite mice were immunized with both human and dog protein X, which has 75% homology on the ECD domain. The flow titer figure shows that at 1 to 100,000 dilution, we can still see fairly strong signal against human protein X. Beacon B cell sorting isolated 567 positive B cells. Among these cells, 352 sequences were extracted and cloned. Totally 308 antibodies were derived. Among which 113 were both human and dog positive. 10 were dog-only positive. These data shows that RenLite mice can generate high-quality hits that are cross-species reactive. To summarize my talk, I have introduced to you our first and second generations of immunoglobulin humanized mouse. The RenMab platform provides fully human-heavy and kappa light chain repertoire diversity and enables efficient antibody hit generation with desirable drug-like properties. The RenMab HITS platform conquers challenging targets and facilitates clinical development by generating species cross-reactive antibody candidates. And thirdly, the RenLite Platform accelerates bispecific or multispecific antibody discovery with common light chain and full human heavy chain repertoire. These platforms promote the discovery of functional antibody hits that exhibit human antibody features and solid drug-like properties. Biocytogen is open for collaboration and partnership on these platforms. To further support our collaborators, we offer antibody discovery service. We can perform antibody discovery in-house using RenMab or RenLite mice and deliver antibody hits to you. So there's no need to find a third-party CRO to perform the immunization and antibody discovery. Our antibody discovery service also opens to the public. Our Beacon Optofluidic system shortens the discovery process from months to weeks. We also have additional platforms such as FACS-based single B-cell cloning and phage display library technology for combined discovery strategies if necessary. Biocytogen also has a vast inventory of humanized mouse models for pharmacology and in vivo efficacy studies. Overall, our integrated platforms can support you at any stage of your therapeutic antibody development process. We welcome all potential partnership and collaborations. Please contact me if you are interested in working with us. Thank you for listening, and I'm ready for questions.